Good.
Yes, we are going live.
Dobar dan. Good afternoon, everyone. Welcome to the panel discussion about how medicines are discovered and developed in pharmaceutical industry as part of the European Researchers' Night in Bosnia and Herzegovina. My name is Ljiljana Đukanović, and I will be the panel moderator today. We will have 45 minutes for the discussion and 15 minutes for your questions. Please, I'm inviting everyone to give us your questions, whatever comes to your mind. Feel free, don't be shy. The discussion part, the question answering part is usually the most fun one. So let me introduce our three wonderful scientists that we have here as our panelists. And if you allow me, I'll call them the rock stars because they're really wonderful women scientists, and I'm so grateful that they're here with us today. So Ozge Luchkan, who is currently working as a project team leader in dermatoimmunology department in Novartis Institute of Biomedical Research in Basel in Switzerland. She got her PhD in the US in molecular genetics and genomics, and then she went to Spain, Madrid, where she worked as a senior scientist at the Spanish National Cancer Research Center. She has a really extensive experience in bone biology, skin biology, immunology, and cancer biology. And Ozge loves science. Ozge also loves life. So if you are Ozge's friend, you'll get to experience witness some incredible cooking. You'll get to travel a lot and laugh a lot. That's basically what I get to do, being her friend. So thank you, Ozge, for being here, and hoş geldiniz.
Bravo.
Then we have Amparo Lago here with us. Amparo has 32 years of multidisciplinary career with experience in drug discovery, drug development, business development, and alliance management, which she will tell us more about. And it's actually alliance management, what she's been doing the last 10 years at GlaxoSmithKline in Pennsylvania, US. She actually is a chemist. She got her PhD in organic chemistry, then went to do a couple of postdocs, including a postdoc at Stanford University. So really extensive, both academic and industry experience. Now, don't get this impressive biography intimidate you, because Amparo is actually a lot of fun, and she'll be more than happy to sit with you and discuss Balkan politics and history over some good Spanish wine. So thank you, Amparo. And
a lot of wine.
Bienvenida.
Gracias.
Alma Sabas. She's Bosnian. She's from our part of the world. She works at UCB Pharmaceutical Company, focusing on clinical drug development, regulatory affairs, and medical affairs. So she's basically responsible for clinical strategies and trials, which she will talk more about. She got her doctorate in pharmacy and postdoctoral training in clinical research and drug development. Like I said, Almasa is from Bosnia, and I want to mention that although she's left Bosnia many years ago, she's extensively working and helping and contributing to the development of our country in many different ways. One of them, just to mention, she's a co-founder of Bosnia and Herzegovinian American Academy of Arts and Sciences. So she's done really wonderful work, and as I'm also a member of the Academy, I have to say she's been very inspiring to me and many other people. So thank you for being here, Almasa, i dobrodošla. Okay, so all right, so I'm going to start by saying something, which is that I want to admit that when I was even a student, and I was a science student, I actually did not really know much about research that was going in pharmaceutical industries. So you kind of think great innovation takes place in university labs, and then pharmaceutical company does the magic and you get your pills in the pharmacy. And I think this is probably still the case that many people think, you know, everything that happens in the pharmaceutical company in terms of research is kind of a black box. So that's why we're here today, to really talk about this process, all the efforts and resources and the processes that go into, you know, from the discovery to the commercialization of a drug. And so basically we'll tell the life story of a drug. And I would like to start with Özge, who's really at this very beginning stages of this whole life, let's call it, of a drug, which is discovery part. So Özge, please tell us a little bit about what this looks like. What is it exactly you do? And also take the opportunity to give us a bigger picture on what your company, for example, you know, some statistics and basic information that we can build a picture of what efforts and resources are put into a research in a pharmaceutical company. The floor is yours.
Thank you so much, Liliana, or teşekkür ederim for your efforts, also to say it in Turkish, and for the invitation to be here. I'm definitely excited to talk a little bit more about this. Just as a disclaimer, I'm here representing myself and not any views of Novartis, the company I work for. Also to say that I'm new to drug development and discovery. I joined Novartis Institutes of Biomedical Research, or NIBR, about 3 years ago. And I am also learning a lot of the process. And as Liliana described, I think coming out of academia, we're not really well trained in drug discovery. And it's an impressive science that requires a big team effort. I think if there's something that you should get out of this session here is that drug discovery and development requires big teams and a lot of money. Okay, so what does it actually take? How long does it take to take, let's say, an idea, a concept, to a drug in the clinic? Today, the average time that this process takes is between 10 to 15 years, which is a really long time. And to put that into perspective in history, I think vaccine development, for example, took around minimum 10 years. So what you're seeing with coronavirus vaccines at the moment is unprecedented and super exciting for the whole world, but also for the scientific community. Okay, so it takes 10 to 15 years. And what's actually the success rate? This is quite a low number. You'll be surprised maybe to hear that only between 1 to 5% of ideas or concepts or targets that start as drug development projects make it into a drug. And for example, Novartis invests about $2 to $3 billion for the development of a drug. So this is, as you can see, a big investment in money, but also people and time. And that it's actually a process that starts with, like I said, the idea generation, preclinical testing, clinical testing. And as we continue in the process, the investment also grows, because often I think Almasa will talk about this, the clinical trial phases are often the more expensive ones, as they have to be multinational and quite complex. So I'll tell you a little bit about sort of the role of a biologist and sort of project team leader, as we call it in Novartis, in this process. Again, I'll emphasize the fact that this requires a team and it's not done by one person. So I, for example, lead a small lab of a few people, and at the same time, I lead a project. So what do I mean by a project? A project starts with basically a validated target. At least in Novartis, this is the case, and I think different companies might have different ways of doing this, but the process in general is pretty similar. So in the beginning, we need biologists, we need chemists, we need computational scientists, we need clinical scientists, and all of these people get together and drive a project forward. So usually, as I said, drug discovery starts with just an idea, and this idea has to be tested and validated in a laboratory. And in Novartis, an idea that is validated is the point of, or let me rephrase, validation is the point where a drug becomes an official drug development project. So the 1 to 2 years before that you spend in the lab is not even counted probably into this 10 to 15 year timeline. So there are many ways an idea or a hypothesis can be born. You could be reading about it in the literature from a paper. You could have found it looking at so-called big data. Data coming from patient samples, or you could be identifying it through other measures. It has been, for example, shown that the chance of success of a drug is doubled when there's a genetic evidence that a gene is associated with a particular disease. So for example, looking for mutations or polymorphisms that are associated with particular diseases is an important aspect of the job. So once we identify the idea and we generate it, we utilize many different techniques in the lab. We start, for example, first in vitro, then ex vivo, and then we go into in vivo animal models to test our hypothesis. And as I said, this takes about maybe 1 to 2 years once you can come to a place where you feel confident that this target or pathway is important to modulate to come to a disease transformation potential. So let's assume that you're very confident that your target or your pathway is going to modulate or transform disease. Then what you have to do is think about it, whether this target or pathway is amenable to drugs, or can it be druggable, in other words. Nowadays, for example, our head of research tries to, or says all the time that there's nothing undruggable. And this speaks to the fact that there are now many different innovative efforts to try to move away from the classical way that we used to do drug discovery. So just to give you an example, if you are working on a cell surface protein, so a protein that's sitting on the outside of the cell, these proteins are often amenable to inhibition, or when I say inhibition, I mean stopping the action of this protein by antibodies. But you have to consider antibodies are large molecules, which means they have to be injected most of the time to people. They are difficult and expensive to develop, and often come with their own issues that for time purposes I won't go into. On the other hand, we have so-called the small molecules or the low molecular weight inhibitors, which are often, like I said, small molecules, so they can be put into pills and they can be taken orally. They're also a little bit more easy to scale to bigger production. However, to find a small molecule that's very specific to a particular protein has its own challenges. In the past, for example, kinases have been a class of proteins that have been amenable to inhibition or stopping their action by these so-called small molecule or low molecular weight inhibitors, because they bind to a particular pocket, often, not always, so-called the active site of the kinase, and they can inhibit the function of these proteins. But imagine you want to inhibit the activity of a transcription factor. What do? You cannot generally make an antibody for it because the antibodies don't go into the cells very easily. And low molecular weight inhibitors are harder to, or at least activity inhibitors are harder to come up with. So for example, one of the new ways that industry has come up with in the last few years, industry and academia as a collaboration, is this so-called targeted protein degradation platform. And this is a very interesting and innovative method. So basically, you are hijacking the garbage disposal of a cell. So when a cell has proteins that are not functional or unwanted pieces of things, it has a way to send these proteins to degradation, and thus they're removed from the cell. And so what we can do now is say, okay, well, instead of inhibiting the action of this protein, what I want to do is tag it in a way that it's sent to the proteasome and degraded and thus removed from the cell. So those are, for example, approaches of innovation that you could think about. And there are many, many examples of different platforms and approaches one can come up with to stop the action or get rid of proteins that you believe are pathological in this process. Now, when you decide that your target is druggable or that you will try to drug it, then you have to come up with an experimental setup, or we call it a screening assay, that is amenable to this process. And why do I say that? The assay has to be easy, it has to be reliable, and it has to be robust. And that's not often easy to find a specific readout downstream of a particular protein. And imagine that in one project for one molecule, you will have to screen anywhere from 500,000 to 1 million compounds to come up with a few series that actually will lead you possibly to a drug. So if your assay is variable and it's not robust, you will not be able to come up with conclusive results. So those are sort of the things that you have to think about as a biologist in the early preclinical work of drug discovery. And I think Amparo can take it up from here and tell us about actually this process from a chemist's perspective and a little bit further down the drug discovery and development.
That was great, Ozge. Thank you. Can you quickly just tell us before we move on to Amparo, what diseases do you work on exactly? If you can share that, just generally, what are you trying to target, or what are you guys working on?
I can't tell you what I'm trying to target, but I can tell you what I work on.
Disease generally, or
yeah. So I'm in a department called ATI, or Autoimmunity Transplantation Inflammation. This is actually one of the largest immunology departments that exist in all of the world and industry. We are about 250 biologists and around 200 other chemists working on immunology projects. And I am part of the dermatology unit, but we have a so-called matrix structure. So just because I'm in dermatology doesn't mean I only work in dermatology. At the moment, I work on diseases such as atopic dermatitis, which is also called eczema, as well as psoriasis. And I also work on joint inflammation, anywhere leading from psoriatic arthritis, rheumatoid arthritis, as well as spondylopathies in general.
Sounds super exciting, really.
It is. It's fun.
Yeah. I'm jealous. Okay.
Thank you.
All right. So let's move on to Amparo, who will tell us a little bit also of that process of drug discovery, but from a chemist point of view, and from when she used to do it, but also touch a little bit on what she's doing right now, which has to do with how pharmaceutical companies go out into the world and collaborate with academic labs or companies and put efforts together to come to productive solutions. So Amparo, the floor is yours.
Well, first I want to thank Ozge for a wonderful introduction that leads very well into what I wanted to. I mean, she's covered the process very well, from early ideas to what we do in preclinical studies. I wanted to give you a little bit of the historical perspective, because there is an enthusiasm that overcomes. When you're young, you want to discover things, you want to do novel things, and you're curious about the process. And this is exactly what is happening right now. And Ozge also mentioned how short was the timeline now for this vaccine that has been discovered, that is built upon the work of many other people. I mean, it's been so fast because so many people have been working on these new approaches for vaccine from before. It's not like everything was discovered instantaneously just because we needed it, and just wonderful that this has happened right now, built on the work of others. And the period of time that where that succinctly Osge has described, it was for me, for my generation, it was revolutionary in the sense that it was what we call rational drug design. Instead of, so this was a novelty, and I lived through this period, and I was part of that discovery, which now is been moving forward and just always pushing the envelope. So I encourage all of you listening to continue pushing the envelope, because this is truly the best endeavor you can push forward. And as mentioned by Osge, as a chemist, what we, in a very old fashioned concept before, when we tried to discover, with scientists or medics in general wanted to intervene on a disease process, they had very few tools. It wasn't understood how disease work. It wasn't understood how to interfere with the disease process. And the whole, from a chemist point of view, everything started almost just by trial and error. Just started with dyes. A big chemical industry started with dyes, like for clothes and everything else. That's what make money. They started testing these molecules, and they discovered they had anti-cancer properties, but there was no rationale behind it. It was just, it seemed like these molecules were toxic. They killed the toxic cells and others, and it helped patients. But what we as scientists discovered, it was that there was a target, as Oscar described, that it wasn't random. There is certain receptors or ways of modulating disease. And as a chemist, we have the ability, and this is something that, as Audrey was saying, we work in big teams. So chemists work with biologists, work with molecular biochemists, molecular biologists, and we as chemists, which is something that is, we read a molecule and we say what parts of the molecule interact with what, and we can tell the biologists. And this is the revolution that happened when during my career. So there is a target. We think that because modifying that target will cause this effect on the cell, which would lead to impeding the disease to progress. And how do you intervene on that? As a chemist, and we work more on small molecules, as Osgi is mentioning. And originally, when I joined the industry, the classical example was all big companies had libraries of compounds. We're talking maybe 1 million compounds. So what we did is, what it was done traditionally was a very trial and error system. You have your assay developed, but that you think it has an effect on the disease, and you basically screen. So you have your target, you have the molecules, and you just span and see which one of those molecules was active. And then the chemist would come in and make it more active, so it would be more effective, less toxic, orally available, so you can take it as a pill. And that was what chemists did. But what happens in the meanwhile, we got more and more knowledge about what that target was, and we could make 3D pictures through modeling, through X-rays, and then we look at what the active side of that molecule would be, and we started designing rather than panning, not knowing anything about it. We could design, focus on what we want to the disease process we wanted to intervene. And Ozzy also mentioned kinases, and I wanted to use that also as an example. Kinases is a very common enzyme in the body, and they transfer a phosphate from just to give energy to the cells, and it's a very common cellular process. And at the beginning, it was thought that this is impossible to interfere because it's so common in the organism, the kinase phosphorylation. But what was discovered is that they always bind the same way, and they have 3 points of attaching to the ATP, and it was with 3 points of contact. and it was always the same 3 ways. But what chemists learned to do was to modify that molecule so there was selectivity. So if you wanted to work on a target, you could modify the area around those points of contact to affect to give selectivity to those small molecules and only do whatever they were we were aiming to do. So that was a revolution that happened as a chemist during my work at GSK. In fact, one of the companies, the legacy company that I worked for, they decided to have, instead of panning this whole universe of molecules, they make like a little library of kinase inhibitors, and you can always find an inhibitor to your kinase. And then the chemistry job will be to take one of these small starting molecules and make it more selective for the target that you wanted to work with. So working with chemists, working with biologists, you make that molecule more amenable to your disease, more selective, so it doesn't have a pan-active effect, and you move on into the clinic. And in fact, most companies have kinase inhibitors that now are used as drugs. In particular for cancer, is a very common start of chemistry for people to go into this disease. So just wanted to particularize, like with an example, how, you know, exciting was the chemistry world when I joined, and one of the reasons why I joined industry from academia. And as Liliana is saying, a company such as GSK or Novartis or any other company cannot have all the ideas in the world, cannot be the be-all and know it all. So many times the basic research is done in academia, because the time, effort, care that a professor dedicates his or her whole life to this pathway cannot be done in a commercial way. So we as industry go to these scientists that are dedicating their life to understanding this pathway and work with them to create practical solutions for a disease, for example, or even technologies. This industry, it could be a small company or an academic that has a technology that would be useful, as Oscar was mentioning, to help us select the best targets, the best molecules, the best models. As Oscar was mentioning, and I don't know how much I'm doing on time. So for example, not just working on one particular example, we talk about cancer, and we want to be the closest we can be to the actual model. We always use, like, you know, we could use like cells, cancerous cells, but that model is very removed from the real model. So there's always ways you can treat animals, but rats are not human. So it's another model that it has this. So every model that you want to get to the real disease has their caveats, but it's the closest we can get. And we're always working to getting closer and closer to the actual model, rather than using the human being, which, you know, we try to spare on these trial and errors if we can. So, for example, one is right now we're trying to acquire as much knowledge as we can, and we're trying to have organs in a chip. For example, what does it mean? You want to have a liver that is not a human, is not a liver for a human, but it's a chip, and you use human cells to make it behave like a real organ. Or you have organs that you grow on a 3D matrix, because that's closer to all those things. Normally are developed in academic groups, or, you know, sometimes even government groups. And this is something that is what we in industry are always looking for, creative ideas that we can take forward in a very practical way. And this is what I've been dedicating my efforts in the last 15 years of my career. So now that we told you about how we do it at the beginning, how we try to make these molecules that are less toxic in animal models or in vitro models or with other technologies. I'm gonna have Almanza is gonna tell you how, when we get to the clinic, what do we do? Oh.
Perfect. I don't even need to do the moderator job. Amparo, you did it perfectly. Almasa, go ahead, tell us about all the large clinical process that now has to happen.
Sure. Thank you. First of all, thank you so much for the kind introduction. I just cannot be more humbled by being invited to this panel discussion. And it truly get a sense of appreciation how drug development is a multidisciplinary approach. To just add on to what Ozge and Amparo has said, there's not a single person or a single body that knows everything about what it takes to bring medication to the market, but it's truly a multidisciplinary approach. And I also want to, before I dive into more technical language, I really want to greet all of all of the viewers out there, whoever you may be, from Bosnia and Herzegovina or from anywhere else. I just send you big hugs to all of my fellow Bosnian Herzegovinians on the other line. Any folks from Donji Vakuf, that's my hometown, and big greetings to people from Jajce, Travnik, Bugojno. Those are very nice čaršije, as I like to call them, that are very inviting. So thank you so much, and I hope you all learned something from here. So again, just to emphasize, yes, I here as well, I'm on my own, opinions are my own. I'm not going to position any view of the company that I've worked for in the past, but really share some of my knowledge, what I have acquired over the years working in clinical or global medicines development. So think about a medication, any medication that you have to take for, whether it's an antibiotic, whether it's a little pill for pain like ibuprofen, right? How many of us have taken ibuprofen for pains and aches? Think about that medication before it was a medication, before you could go in and get it from your doctor. What is the life cycle of the process of the development? So for any molecule, to what Amparo and Ozge have said, before any molecule can be truly a medication, it needs to go through such a rigorous process of development. And after it's been studied in animals like rodents, for example, or rhesus monkeys, or just that preclinical research and toxicology, then what we would do is take that medication through 3 stages of development, or that potential medication through 3 stages of development. The first stage is called phase 1. So after the medication or the entity, molecular entity, is decided to be developed, then that medication will be tested. First in humans, healthy volunteers. So what do I mean by that? So it could be usually young individuals, male or female, that have no underlying diseases, that are fairly healthy. And we want to see and observe how that molecule behaves in the body. So in other words, if you take a pill or a tablet or a syrup, how it is absorbed in the body, how does it reach that target, that area where it needs to act. So first we will do what's common to do is a set of pharmacokinetic studies in in healthy volunteers. So that would start with looking at how this drug behaves in the body, how it's absorbed. So when it reaches the stomach, what is the absorption? How it is distributed within the vascular system, for example, or how it's distributed in the blood, and also which organs does it go to? Will it concentrate more, say, in the liver? Will it go to kidneys, or will it go to bile? You name it. So what's often done is you observe how that medication acts in the body, how it's absorbed, how it's distributed. Big one is how it's metabolized. Everything has life, so we have to be mindful of how it's metabolized and which organs will metabolize that medication. And then how it's excreted before you have to take a new dose. Before it can be taken into a next stage of development, the drug needs to be safe. So in other words, there should be no serious adverse events or severe adverse events, any trends in safety that could be a red flag for a concern, right? You could have the most amazing medication that does what it needs to do, but if it's not safe, it will not be approved by health administrators, health organizations, health authorities. So once that's done, then often the next stage of development is the so-called proof of concept study. And I particularly like the proof of concept studies and the phase 3 studies that will come later. And this is where a medication is tested to see does it really have an effect, that objective effect on the endpoints that are put forward prospectively. So in other words, if you have a medication to treat blood pressure, or does it truly lower blood pressure, and how does it lower blood pressure? If you have a medication for diabetes, does it lower blood glucose? If you have a medication for cholesterol, does it lower cholesterol? And you look at your medication of interest, compare it to placebo or standard of care. In other words, what is currently the standard of care in medicine, in medical practice. And then the studies, clinical studies, are designed as such that they want to show in an objective, statistical way that this potential medication has some effect, and you have to test it. Sometimes you have to test it in several studies to show that your findings are not by chance. So that is, in a sense, what goes on in the phase 2 or like proof of concept studies. And what I like about the very last stage when you think about the development is this phase 3 trials that enroll the diseased population, very large numbers of patients, and you truly test the medication as a pivotal in a pivotal efficacy and safety study before you, as a company, a company can seek a regulatory approval from agencies like the United States Food and Drug Administration. You often hear that as FDA, which is often now in the news. You probably hear that a lot because of the vaccine development. or another regulatory body that is, for those of you in Europe or European Union, is the European Medicine Agency. The particular committee that deals with the approval of medications in Europe is the Committee on Human Use, Committee for Medicinal Products for Human Use, CHMP. So the other regulatory bodies that are often prevalent on the East, in the Asia Pacific, is the Japan Pharmaceutical and Medicinal Product Administration, as well as the China FDA that recently renamed itself. So those are big regulatory bodies that will look at the medication to see if it's truly safe and effective before it can be approved for their citizens. So in a nutshell, you see that development goes on for many, many years. As Ozge had alluded to, this could go on for 10 to 15 years of development. And in the case of vaccine, it is not quite the same as, say, a drug development, but it's very similar, I must say. It's very similar. You have to find that the vaccine would produce some immune response. You have to find the right dose, and then you have to test it in the disease population to see if it would really prevent that disease. So there are some overlapping areas between the vaccine development and the drug development. So just think about, just to leave you with, with a little nugget of wisdom, is next time you have to take a medication for whatever, you have to take it, even for acute indication or for chronic indication. Just know that it has gone through a rigorous review process, making sure that you don't have severe adverse reactions or some serious adverse events that could be more detrimental to patients than be beneficial as side effects. So just think about that next time when you have to take a different medication. But yes, and then once when it comes to medical affairs, that's when we look at different. Just to wrap it up for the last few minutes, just to look at, in a way, how it can be differentiated for a patient, for a medical community. Not every medication will act the same in everyone. It's not a cookie cutter approach. So you will have a different section or different medications to choose from for a patient, and then that's something that physicians or the medical community would evaluate using their judgment, what is best for his or her patient. So it gets to be very interesting.
And Almasa, thank you so much. Wonderful way how you walked us through this essentially complex process. But just a question, maybe you can quickly address. What I find interesting is that actually companies continue studying drugs once they are marketed. So it's not like the research process ends once the drug is in the pharmacy, right? So you continue doing studies and addressing different aspects, or maybe for different indications or so on. So maybe just a half a minute, because we're already getting some questions. So after that, I'd like to jump into the question.
Sure. Now you touched upon an important topic. What happens when a medication is marketed? Then you perhaps want to study that medication in different disease or for a different indication. And that's something I also didn't talk about, is pediatric drug development. Think about the pediatric drug development and the diseases and illnesses that often affect the pediatric population. It could be even like epilepsy. Then you have to study the medications in children and adolescents and perhaps even neonates. So it gets even more complex. So that life cycle management, as we often call it, life cycle management, always happens until maybe the patent expires, or patent may be extended depending on the certain geography where intellectual property can be maintained. So there's all sorts of life cycle managements that could happen. You have to think about this strategically. You have to think ahead what medical community may need, what patients may need. It's always, as Amparo said, pushing the envelope, keep pushing the envelope. So that's how I see it in drug development as well. We keep pushing the envelope.
Wonderful. That's a nice phrase to close the discussion with. Keep pushing the envelope. Okay, we got a couple of very interesting questions. The first one is, what are concrete examples of something being druggable or non-druggable? I believe Ozge was mentioning these concepts. I don't know if she wants to take up on this question, but as you wish, all of you can maybe discuss it. We have 10 minutes.
I can start with that. Thank you. It's an excellent question, and I hope you can hear me because my neighbors just decided to drill upstairs. So what is considered a druggable or an undruggable molecule? So I think Amparo can talk more to the history of this, but generally proteins that have enzymatic functions, such as kinases, are considered sort of easily druggable proteins because they have a particular pocket, as I mentioned, and as Amparo alluded to, for example, for kinases, that we know their binding modalities, and we can screen and then work with the molecules to make them more specific or selective and less toxic. Now, some examples of that are, for example, for many liquid tumors, we have drugs that are kinase inhibitors. Other than kinases, for example, ion channels are, again, because they have enzymatic activity, those are often druggable. Soluble molecules such as cytokines, inflammatory molecules, are quite druggable by antibodies. As I mentioned, receptor molecules on the surface of cells is quite druggable. What is undruggable are basically proteins inside a cell that don't have a particular enzymatic action, like a transcription factor, like a regulating protein, like a scaffold protein. Often these are large proteins and they are very flexible, so it's very hard to find a molecule that inhibits their action. For example, I can give an example that if you wanted to inhibit the interaction of protein A with protein B, we call these sort of the inhibitors of protein-protein interaction. These are considered to be quite difficult to do. They are challenging, the assays are challenging, but also to find selectivity is very difficult. But I think these are the places where we're starting to create more innovative platforms to think about how to inhibit these more complex structures. Maybe Amparo can add more to this.
Well, actually, you put an example that I was thinking about. I think we have a connection here, and I like Almanza's pushing the envelope, because actually what we think today is not what is going to happen in a little while. And exactly. I can give you an example of actually a drug in the market that actually is a protein interaction that was supposed to be impossible to do, because you imagine you want to stop an effect to what we basically are saying in the old model is to put it like a very simple language. We always thought what Osge is saying. We have a key and a lock. It's a very old concept, it doesn't work. I know the protein changes, but let's say if we put like wax in that keyhole, then when the molecule wants to get in, it can't, because you already block it. So that's why we were trying to interfere with the disease process. But if you cannot put wax into when two proteins come together, and what do you do with that? And that's why we, for a long time, we thought that was undruggable. But actually, erythropoietin, which is something that is in the market, Promacta is called, I think, or whatever. So it demonstrated that a small molecule is it's the only one I know, actually. So it's not like it's a general, but a small molecule was able to impede this process. So people that have problems when they heat, when they have little bumps and they bleed to death, they can take erythropoietin and manage this disease. So what we think today, what I'm trying to say is that we're counting on you guys, young people, to push that envelope, go forward, because what we thought it was impossible yesterday, like who would have thought we had a vaccine in a year? Well, it's because many people work on that, and we thought we couldn't do these protein-protein interactions, but we try and we manage. So don't think about the limitations. We're counting on you to push the envelope and tell us you were wrong. That is not, you thought it was non-druggable, but it is, because, and that's the piece of science that needs to be discovered. We don't know everything. We need to find out more.
Yes, innovation is key.
Nice, very inspiring. Thank you, Amparo. I think we have a question here for Almasa. Could be a good closing discussion, since we have 7 more minutes. What do you think about the extremely fast development of COVID-19 vaccine? Is this a leap forward in drug development? I think maybe Almasa can address this.
Yeah, thank you. That's a good question. Certainly, this is the first time in history that a vaccine has been developed so soon, so quickly. Usually when you think of drug development, it has some risk as part of it, as part of the development, as far as when to initiate phase 2. You want to wait for results of the phase 1 or wait for results of phase 2 before you go into phase 3. And certainly with the production of a vaccine, for example, or medication, a lot of these steps were done in parallel. And this is the only way to expedite it. So rather than waiting for, say, phase 1 trial to finish, and then at the end, like you'd want to maybe start phase 2, and then the phase 3, what you see here is like pretty much phases stacked upon each other. And I'll take even Pfizer as an example. They have started making the vaccine at risk. Just imagine the cost and the huge financial risk that and even scientific risk that these companies are taking. But this is unprecedented. It is truly is a leap in how we develop drugs, but that is not to say that safety will suffer. I have confidence in Food and Drug Administration that they will review all the safety data, and there's an advisory committee coming up next week, which I think all eyes will be on that advisory committee. And then in the week after that, there was another advisory committee for the Moderna vaccine. So I have full confidence that in our regulatory system, that they will not jeopardize that integrity, that scientific integrity, and not cut corners on that. So yeah, this definitely is a leap in drug development, vaccine development in medicine, for sure. Quite remarkable.
I guess what this vaccine development showed, and perhaps we were not aware of it fully, is that we have so much knowledge and technology, sort of logistical power to make a vaccine so fast that, you know, this demonstrates that. I think maybe people don't realize, you know, the extent of resources and the extent of knowledge and capabilities, technological capabilities that we have. And that's really the reason why this could happen so fast, rather than rushing things and skipping steps. No, everything happened as it usually happens. But, you know, when in 2020 we have incredible amount of capabilities. So that's definitely, I agree.
An incredible need, which has pushed all of this. And
yeah.
So this is extraordinary times. But as you know, important to say exactly that safety should not be sacrificed for this process. With the investment had caused a novel technology, RNA, which never used for vaccines, to be developed to this level so quickly, and then all the investment has compressed these timelines.
And I think we have to also give credit to all the agencies that have really expedited timelines. Things that had 10-week, 12-week turnaround times have now shrunk to 48-hour timelines. People have worked night and day. So I think that the people power on top of of course the knowledge and the technology that was there anyway, and the way it's been put to use is incredible, in my opinion.
Yeah, I couldn't agree more. I think there's an army, a true army of people, whether it's in companies or health authorities, that are working not days, but they're working evenings and weekends. Yes. I am confident about that, and my kudos to all of them, because science will win.
Beautiful.
Wonderful. This is amazing. We do have a couple of minutes left, so I would like to ask each one of you to give some last words. Now, I'll leave it up to you. Perhaps, you know, I'll give you some ideas. Maybe you want to, you know, say a few words on encourage women scientists to go into science and research, perhaps in pharmaceutical companies. Or anything you want to say really regarding what we've discussed, or the COVID situation, some last words of optimism and motivation, let's say. We start with Özge, the same order.
Okay, no pressure.
You're like on my screen, so.
Okay, well, where should I start? I have to say, I think for me, science is not just a job, but it's a passion. And research is something that excites me. I wake up every morning excited to go to work, or now stay in my home office, and bring some knowledge forward, right? And I have to say, I come from a very small country, from Cyprus, where not many people get the chance to go outside, especially as a young female, to move away and explore. And I really want to encourage everyone that if you have the curiosity and if you have the will, go explore, travel. You meet incredible people within the scientific community. Liliana and I met through science, and our friendship has continued, extended beyond science, and continues to do so. So I think science is more than a job, it's a passion, and if you have the curiosity for it, nothing should stop you.
Well, what I want to tell you is that I am, if you have to work, you better choose a work, type of work, if you can, that engages you and that you feel that you're contributing, and in all the time. And it's a game of patience, but also all the time that I've been working for these many years, I can tell you that I felt motivated, that my contribution, when looking overall, is something that keeps me coming back to work and looking at what other things can be accomplished. It's a very motivating thing to have a career that you are inspired to do well, and to have a, you know, to have something that at the end of the day you say, yeah, I moved, maybe today was a small step, but maybe I'm moving things forward for people to feel better eventually, although my contribution is here early on, but like we were saying today, there is a larger picture, and it engages your brain and engages your imagination, and it's something that is something that otherwise your job becomes, you know, too routine to keep this motivation for a long time. So I encourage everyone that has curiosity and desires to investigate like a novel, what, you know, mystery can be uncovered, to think as science as a possibility.
Yeah, and I echo what Amparo and Ozge have said. Really would love to encourage everyone, especially young girls, to stay in school. And also with the younger generations now, with the sustainability being so permeating in our lives, and think about how we want to, what kind of a footprint we want to leave on or live on this earth. Think about the climate impact, because we know that it has been proven over and over again that the more if we keep living the way we live, the way we treat the planet, more pandemics could come. So some experts are even saying that perhaps coronavirus could be even just a dress rehearsal for what else could come. But again, our mind, our curiosity, our knowledge, and just make sure you surround yourself with those who think differently, or even just have questions, just ideas that you could pressure test with someone. It doesn't have to be on a big scale, but think about what kind of an effort, or what you could do to maybe not change the world, but just change the environment, change the street maybe that you live on, or change the town to where you live, or just to surround yourself with people who perhaps think a little bit differently. You don't necessarily have to agree, but that diversity of thought will create something powerful. And just stay in school, be curious, be engaged, don't mock people's ideas, because you never know what could transpire from them. And just stay positive and think forward, not backwards.
Wonderful. Wow. Well, Ozge, Amparo, and Almasa, thank you so much for your time, for your knowledge, and for your wisdom. This was really, truly wonderful. I also want to thank all people that have been watching us, and special thanks to the organizers of Researchers Night. This has been an amazing team that's really bringing forcefully science to the Bosnian public, and it's amazing. And yeah, I think I will leave it with a sentence that I see here in chat that says, "Science will win." So let's leave it at that. Everyone keep safe, and hopefully the situation, the COVID situation, will be resolved soon. So thank you so much again, and
Thank you for the opportunity. Thank you.
thank you
for inviting us.
Bye-bye.
Bye.
Ciao.
Bye.
Everyone stay safe.
Bye.